In this section, we're going to study second messenger systems. And in most cases, second messengers are brought about through a cascade of events that act through G protein coupled receptors. And what we will see is we have what's called a first messenger. In this case, our first messenger is going to be a hormone. We have seen second messenger systems in previous chapters, especially when we looked at the nervous system, the autonomic nervous system in particular, and also when we looked at the signal transduction of light in the eye. So what we're going to do now is we're going to consider them in the context of hormones, but in either case, be it a neurotransmitter that diffused across the synaptic cleft or a hormone that's circulating in blood plasma, the first messenger that's going to kick off the cascade of events that will lead to the production of second messengers within the cell, the first messenger is going to bind with a membrane-bound receptor on the cell. So in this case, it is a transmembrane receptor in that it is a protein that spans the membrane and has a binding domain on the outside surface of the cell. And on the inside surface of the cell, we see that the receptor is coupled to what we call a G protein. In the inactive state, the G protein is bound to the intracellular side of this membrane receptor. And it is held in place by this molecule of GDP, which is guanine diphosphate. And remember, GDP, GTP, is very similar to the nucleotide ATP, or adenosine triphosphate. So remember, if you have 3 high-energy phosphates on your ATP, you would also have 3 high-energy phosphates on your GTP, and then when you lose one of those phosphate groups, then you have 2 phosphates left. And usually you give up one of those phosphates to yield energy, as we have seen in pathways for cellular energy and metabolism. So now let's look at this second messenger system, starting with the first messenger, which in this case, for our purposes now, is going to be a hormone. This hormone is going to bind to its receptor. And this is the first step in this creating the appearance of second messengers, and that is the binding of the first messenger to the membrane receptor. Once this happens, then that is going to cause a change in the G protein so that it is now going to bind a high energy molecule, GTP. So it's going to exchange GDP for GTP. And when it does so, that's going to allow this to dissociate from our membrane receptor. And now the G protein is going to activate, in this case, it's going to activate a membrane-bound receptor called adenylate cyclase. And so here's our GTP, and it still has its guanine triphosphate, which is a high-energy molecule that's bound to it here. And as long as that's bound to it, this thing will stay active. So, sorry, it's our G protein here. So our G protein dissociates from the receptor and is going to activate this enzyme, in this case, called adenylate cyclase. And adenylate cyclase is an enzyme that will take the high-energy molecule ATP. And I'll put it up here as a high-energy molecule because we know what it does in the cell. But this will cyclize it into something called cyclic AMP. So cyclic adenosine monophosphate. And this now is our second messenger. So our second messenger in this case is cyclic AMP. And cyclic AMP can go all over the cell and activate all kinds of enzymes. One of the enzymes that it's going to activate is called a protein kinase A. And what does a protein kinase do? A kinase is an enzyme that adds a phosphate group to something. So this kinase can go and do what we call phosphorylate other things in the cell, like other enzymes. And so some of these enzymes, once they are phosphorylating them can turn them on or turn them off, depending on the enzyme. But basically, this is going to change the activities of the cell, the enzymatic activities of the cell. Cyclic AMP can also cause increased gene transcription. So we can actually have a change in effect of gene transcription once cyclic AMP is activated. And as I say, it usually activates this protein kinase A, which can go all over the cell, phosphorylate other enzymes. Another thing it can do is it can phosphorylate channels by adding a high energy phosphate to them. And that can cause channels to open or to close. So we can open channels intracellularly by phosphorylating them. And basically what we see happening is we've got a whole bunch of cyclic AMP being created as long as this G protein is causing the activation of adenylate cyclase. So we can have one binding of a first messenger. So we could have a single molecule of hormone binding to a receptor, and then that activates this G protein, which activates adenylate cyclase, which will continue catalyzing the conversion of ATP into cyclic AMP. So this creates hundreds, if not thousands, of second messengers in the cell, and cyclic AMP, once it has activated protein kinase A. Protein kinase A can go all over the cell, phosphorylate other enzymes, it can change gene expression, it can open channels, it can do all kinds of things within the cell that changes the intracellular activity. So this is one way that second messenger systems can operate. Now, interestingly, I have shown you the second messenger system that operates through adenylate cyclase. Let's say, for example, that instead of this activating adenylate cyclase, the G protein instead, and some will do this, would activate another enzyme called phosphodiesterase. Now, phosphodiesterase is an interesting enzyme. There are what we call cyclic nucleotide phosphodiesterases, and I'll just abbreviate it PDE for phosphodiesterase, and that does the exact reverse. What that will do, instead of cyclizing ATP into cyclic AMP, it will convert cyclic AMP, an active second messenger, it will convert it into just AMP. So it deactivates our second messenger. So instead of creating second messengers, it basically deactivates them. So depending on the receptor type and what the G protein does, what it activates, be it adenylate cyclase or phosphodiesterase, the cellular activities are going to change because cellular activity is modulated by cyclic AMP. And typically cells have a balance of enzymatic activity that regulates the amount of cyclic AMP in the cell. So we could have a first messenger bind to a receptor, causing the G protein to activate phosphodiesterase, and that would convert active cyclic AMP molecules into just plain linear AMP, adenosine monophosphate, and this is not active. So this is not going to cause all of these changes within the cell. Now, eventually, this activity will cease when finally the enzyme here, I should say the G protein, will catalyze the GTP into GDP, and then it will then basically reassociate with this membrane-bound receptor. By this time, the first messenger has dissociated from the receptor. This will reassociate with the receptor until the receptor is again activated by the first messenger. So in this case, we've seen how a first messenger can either upregulate the production of cyclic AMP in the cell by causing the G protein to activate adenylate cyclase, sometimes also known as adenylyl cyclase. Or, in some cases, the G protein can activate phosphodiesterase instead. And instead of creating cyclic AMP, it basically deactivates the cyclic AMP. And that causes all these downstream metabolic processes in the cell to shut down or to be diminished. So that is one of the cyclic, or I should say one of the second messenger pathways that we'll be looking at, and that is related to the production of the second messenger cyclic AMP. The second one we'll be looking at is related to the use of calcium as a second messenger. Now let's look at a different second messenger pathway that involves calcium as the second messenger. So in this case, we're going to have, as before, our first messenger, which is going to be our hormone. That is going to bind to our receptor. That's going to cause the G protein to exchange GDP for the high energy GTP. And that's going to cause this thing to dissociate. And when it dissociates, it will activate an enzyme called phospholipase C. And I'll just write that as PLC, phospholipase C. And phospholipase C is an interesting enzyme because, as the name implies, it will cleave a phospholipid. And as you recall, the cell membrane is made up of phospholipid. It's a phospholipid bilayer. So, just as a quick reminder, you probably remember the general structure of a phospholipid. The phospholipid is basically built around the glycerol molecule. That's this molecule right here. The glycerol molecule, and you have two fatty acids that are attached to it. And so at the core of this thing, we have what we call a diglyceride. And then we have a phosphate group. And the phosphate group is simply a phosphate with some other moiety attached to it. In this case, we've got carbon ring with some more phosphates on it. And this is just sort of a very generic phospholipid. And when we look at what phospholipase C does, it is going to cleave the phospholipid off from the diglyceride. And we also sometimes hear the term diglyceride, diacylglycerol. Well, the particular phospholipid that is cleaved is called phosphatidylinositol bisphosphate. Now, am I going to make you remember that? No. We abbreviate it PIP2. But in any case, this phospholipase C is going to cleave one of these phospholipids, that is PIP2, and it's going to have a phosphate group out here. This is PIP2. It's going to cleave that into a diacylglycerol. That means that little phosphate head of it's going to go off of it. Oopsie. And I'll just abbreviate the other phosphate groups like this. So these are other phosphate groups. I just don't feel like writing them out. And so now this is something called inositol trisphosphate, or IP3. Sometimes you hear it inositol triphosphate. But in any case, the offshoot of this G protein being activated is it will activate the enzyme phospholipase C, which then cleaves PIP2 into this inositol trisphosphate and something called diacylglycerol, or simply just leaves the diglyceride behind. So PIP2 is no longer PIP2. It has been cleaved into a diglyceride, that would be this guy here, and this inositol trisphosphate. Inositol trisphosphate is going to go to the ER, and it is going to release calcium from the internal stores of the endoplasmic reticulum. Now, as you probably recall, the endoplasmic reticulum is kind of like the factory in the warehouse for the cell. The rough endoplasmic reticulum makes proteins. The smooth endoplasmic reticulum is going to make things like lipids and carbohydrates, but it's also a storehouse for other things, including ions. And one of the ions is calcium. So the endoplasmic reticulum has a pretty high store of calcium. So this is going to cause the release of calcium from the endoplasmic reticulum, and also as an offshoot of this activity, we're going to open the cell membrane itself to calcium from the extracellular fluid. So calcium will come in both via the cell membrane and via the endoplasmic reticulum into the cytoplasm. And here, calcium is a second messenger that kicks off a whole cascade of events in the cell that can range from all kinds of things. One of the things that we will see happens is diacylglycerol will convert another enzyme, or I should say activate an enzyme called protein kinase. And we know what protein kinases do. They phosphorylate things. Protein kinase C. And protein kinase C is important because it is also an activator of enzymes. So this whole pathway is going to kick off a whole cascade of events in the cell. And calcium is one of the most important offshoots of this, and that is going to be our second messenger in this case. Second messenger. Sometimes calcium will bind to an enzyme called calmodulin. And calmodulin is sort of an enzyme that has 4 binding sites for calcium. I'm not going to make you know that, but calcium will combine with calmodulin. And here again, we will have increased activity all through the cell, calcium being the main second messenger. So these are the 2 pathways that we're going to study in a lot of detail, or I should say that are responsible for a lot of the things that we see in cells. So the second messenger systems, a lot that we're really focusing on are the cyclic AMP second messenger systems and the calcium second messenger systems. So really this all seems fairly complicated, but when we think about it, what we will see is that at the very basic core of any second messenger system, through G protein coupled receptors, what we're going to see is this cascade of event that can be broken down very simply. I'm going to go ahead and erase this phospholipid here. That can be broken down very simply into a relay. And the relay goes something like this. So here's our relay. We've got our first messenger. First messenger, in this case, is our hormone. First messenger binds with our receptor. Remember, this is a membrane-bound receptor. It's a transmembrane protein, and the domain that binds the second messenger is on the external part of the cell membrane. The receptor then is going to activate a G protein. The G protein then is going to activate an enzyme, and the enzyme will then activate our second messengers. So this is basically the cascade of events that we will see in any second messenger system. So in our cyclic AMP second messenger system, the enzyme is either going to be adenylate cyclase. Some people say adenylyl cyclase. You can hear either way. Adenylate cyclase. And this is going to increase cyclic AMP production. And we're going to increase cyclic AMP. In some cases, the hormone may want to ramp down the activities of the cell. So instead of the G protein activating adenylate cyclase, it will instead activate phosphodiesterase, PDE. And if it activates phosphodiesterase, then that will cause a decrease in cyclic AMP. So that's our cyclic AMP second messenger systems in a nutshell. If on the other hand, we're looking at calcium second messengers, then the initiating enzyme in this cascade is phospholipase C. Phospholipase C, as we know, is an enzyme that cleaves a phospholipid. So it's actually going to cleave a component of the cell membrane into a diacylglycerol, another word for just a diglyceride, which we've seen. And this phosphate molecule, this phosphate-containing molecule, which in this case is going to be inositol trisphosphate. Sometimes you can hear it as inositol triphosphate. In any case, we will abbreviate this as IP3. So IP3 and DAG. And these are going to cause an increase of intracellular calcium. And intracellular calcium is our second messenger. So this is, in a nutshell, the order of events that we would see in a second messenger cascade. Now, as we've seen, because we have a single first messenger binding, we can have an amplification of effects down here, because through each of these steps, we're going to have our first messenger bind to our receptor, and that receptor can kick off a whole cascade of events through one G protein, can activate lots and lots of enzymes. with lots and lots of these byproducts that will go off and create huge amounts of second messengers in the cell. So be it cyclic AMP or calcium, we're going to have a lot of production of second messengers with a binding of a single first messenger. And this is what we call an amplification effect. And the thing about this effect is it can last for long periods of time. So once we've had one first messenger bind and we've kicked off this cascade can run for a little while before it basically settles back down to our starting state. So this is what we mean by an amplification. And as we've seen, second messenger systems tend to operate over the course of seconds, minutes, sometimes even weeks, because sometimes we will see that this will change gene expression as well. So this sometimes takes days in order to get really started, but the second messenger systems will have an effect that is a long-lasting effect. So this is one of the other take-home messages. We have amplification, which is due to this cascade of events where one first messenger can cause the creation of many, many second messengers, and that can have prolonged effects in the cell that have a long time course of operation.